Hey kids, it's Mr. Ortlieb here. I hope everyone is well. We are closing in on the end of the year. Um, just a little update. We're going to end the year a little early this year. My final grades are due on June 17th, so that gives us about 3 more weeks of work. So I'm going to give you a few more assignments. I'm going to leave them open until the end of the year. And hopefully, obviously, I have to get my grades in before the 17th, so they have to be done before then. But I'll give you some more updates on that. But for now, I hope you're not too, your anxiety levels aren't that high, and I hope that you and your family are healthy. Okay, so we are going to do a little lesson on mutations today. But let's kind of start of what we should know about DNA. Now, I just want to keep in mind, I'm not teaching you 100% of what you should have known for the regents. I'm trying to do the stuff that I think that you can handle at home. Okay, so I'm going to try to keep this simple, and let's start with what we should know. Okay, so let's start with this picture here. This picture, you see a cell. You see this little purple thing here. That's the nucleus. Uh, those little X looking things are chromosomes. We should know the chromosomes do contain DNA, and that DNA has these letters in it. They're called bases: A, T, G, and C. G pairs with C, A pairs with T. Okay, so this is the first slide I want to talk about. We should know how DNA is organized. All right, that being said, let's do the review. DNA is organized into chromosomes, and these chromosomes have sections called genes. DNA is located in the nucleus of a cell. DNA contains a code made up of bases. These bases are letters, the letters A, T, G, and C. A pairs with T, and G pairs with C. The sequence or order of these letters is a code to make proteins. So that's one of the things I really want to drive home. We're kind of keeping it simple for you. So just know that the code of letters inside DNA is almost like a code or a recipe to make proteins. Now, why are these proteins important? Because these proteins determine your traits, your hair color, your eye color, the way you make enzymes, your immunity. So all of these traits that we have are are proteins, are based on proteins that are encoded in the DNA. So once again, DNA is a code or recipe to make these proteins, which make your traits. All right. So here's a little mutation. Here's a two-faced pig. You've probably seen some mutations in the past. Um, Two-headedness is actually not that rare in the animal world. It does happen in many species, but it is a type of mutation. Here we have a frog with an extra set of legs. In humans, it's very common for boys, particularly, to be born with an extra digit called polydactylism. Some people are born with an extra finger. Most of the time, it's cut off at birth, but nonetheless, these are kind of common, not very common, but they do happen in species. And this is one of my favorite ones. But I think he was bioengineered for all the X-Men fans out there. All right. Anyway, here's an example of a mutation. Now, mutations can be helpful, they can be hurtful. Here we have a mutation. Let's say you have a mutation with a longer beak. That longer beak will help you get more food and will help your survival. So, mutations can be beneficial sometimes for species. Here's a couple of other mutations here. This is actually a black jaguar. You can see it's got the spots, but it is dark in color. Some scientists have linked the black color in jaguar into immunity advantages, that they have a stronger immune system. So there are some advantages to mutations sometimes. Down here, this is a San Francisco garter snake. Is very, very, very colorful. And what scientists believe is that it's very, very colorful, so it kind of warns other predators not to eat it. Kind of lets predators know, think that it's poisonous. It's not really poisonous, but it's there to kind of pretend or mimic another snake that if you eat it, you might die because it might have some toxins in it. But it's there to protect you. That's why this mutation works for this particular snake. In humans, we all have this thumb, okay, and it's called an opposable thumb, and it gives us great advantages over other organisms. We can hold things, we can throw things, we can give you a thumbs up. No, I'm just kidding. But it gives us an advantage in carrying objects and throwing, especially when early humans were hunting. And here you have this precision grip with a tool. You can actually use a tool to do a job. But our thumb, our opposable thumb, was a mutation, and it has lasted in our species because it gives us such an advantage to survive. Okay, so let's talk about these mutations. What are they? A mutation is an alteration or change in the DNA base sequence. Now, that base sequence are the letters A, T, G, and C. So when those letters get mixed up, they can change a function of a protein, and we'll go through this. But think about it as this. If you misspell a word, it might not make any sense to somebody. If you leave out a letter in a particular word, it's not going to make sense. And that's kind of what happens in a mutation. It changes the letter sequence. So let me give you some examples, okay? Let's start with the causes of mutations. Why does DNA make mistakes? Well, DNA is constantly replicating or copying itself, and it also is doing this protein synthesis. It's a code to make proteins. And in that process, mistakes can be made. It's kind of like when you're copying someone's homework. I know none of you do that, but when you're copying someone's homework and you leave out a sentence and your paragraph doesn't make any sense. Okay, so what it is random mistakes that you can make. DNA didn't want to do it on purpose, but it's a random mistake while the DNA is replicating or copying itself. All right, so what causes these mutations in DNA? Why does it make mistakes when it's being copied? All right, some common things. Radiation is big. Okay, ultraviolet light, number one cause of mutations that cause skin cancer. X-rays, you can't get an X-ray when you're pregnant. The X-rays can damage the DNA of your baby. So forms of radiation can actually cause mutations. Chemicals. Now, what are some chemicals? Cigarette smoke. Many of us know that cigarette smoke causes mutations that can cause cancer. Asbestos, that is another cancer causing material. Things like mercury and heavy metals, those things can cause mutations in your DNA. Now, these things are referred to as mutagens. They're environmental factors that cause a mutation. Radiation is a mutagen. Certain chemicals, like cigarette smoke, they're mutagens. They're able to cause mutations in DNA. All right, another important question. Can you receive a mutation from your parents? And the answer is yes, but it must be in their gametes. After all, your parents made you with their sperm and egg, respectively. And if the mutation is found in those sex cells, you can actually get that mutation. It happens. And I'll give you some examples. Um, it cannot be given to you if the mutation is in their body cells, skin cells, lung cells, whatever. Okay, bone cells. The only mutations that will receive those mutations, the only cells that will receive those mutations would be other cells. So if a skin cell has a mutation, it's only going to pass it to other skin cells. If a lung cell has a mutation, it's only going to pass it to other lung cells. However, it's in the gametes, the sperm or eggs, it can be passed to offspring. All right, so let's get into this. I'm going to try to keep it simple for you. I know learning about mutations is not on everybody's priority list right now, but we have to do what we have to do. Okay, so mutations. So here is a sequence, single strand of DNA, and it's got a bunch of G's, A's, T's, and C's in it. Okay, so this represents a gene, which is a section of a chromosome made up of bases. And this one has 15 letters in it, 15 bases. Okay, so um, what you need to know, which is something that we really don't want to go into because it's a little difficult to understand, and you really need a kind of me standing over you to kind of get it. But I'm going to teach you the basics. This is a code, all right? So this is like a sentence, and it's read every 3 letters. So you take this piece of DNA, you divide it up into 3 letters, this gene, so to speak. You divide it up into sections of 3 letters, and every 3 letters is called a codon. Now, what's special about these three letters? Well, they determine what one amino acid of a protein will be. And I'll show you some examples in a second. Okay, so the first thing you need to know is that the DNA must be divided up into three letter sections called codons. I. Okay, now the first type of mutation is called a substitution. Now, you know what a substitution is. A substitute teacher. Teacher comes in for another teacher. A substitution on the soccer field or football field. A player comes in for another player. So it's when one letter is replaced by another. So here in this example, I take a G at the end of this sequence here, and I made it an A. That's called a substitution mutation, okay? Taking one letter and changing it for another. Now, why can that be significant? Well, let me kind of decode this picture here. So here are all these different letters, and I know I'm looking at it. I see it's RNA and not DNA, but that's okay. That's all right. So what I wanted you to know, so if you take a look at these little boxes here, these all represent amino acids. And the DNA will actually code for these amino acids depending on what the letter sequence here. I know it has a U here. Don't worry about that. Just pretend it's a T. All right. So what happens is that you can actually change the amino acid. So let me show you this one. See this gly? It turned into a sir. All right. And that was because of a mutation. So mutations can change the protein that they code for. All right. Now, there's two more types of mutations that you need to know. The second one is a deletion mutation. And deletion, just like the button on a computer, it means you remove one or more of the bases. So in this case, I removed a base. Let's see if we can figure out which one I removed. I think, oh, right here. The T is up here, but look down here. I deleted it. Now, what's important about this is that it changes those three-letter codons. This is no longer a TCT, it's a TCA. And look what happened here. This ACG got moved over, and now it's GCT. This is TAA, now it's AAT. So it changes the sequence. So mutations can change the sequence of DNA, which can be bad. Sometimes it doesn't matter, but sometimes it can be very, very bad. So in this case, this is called a deletion mutation. All right. There's one more mutation that you need to know. It's called an insertion. Sometimes it's called an addition. I do have addition in the definition here, but it's when you add one or more bases. Okay, so deletion, you take it away. Insertion or addition, you add one. So here we have GAA, nothing changed there, but I inserted an extra G here. You could see I inserted it between the T and the C. And now it's changing the sequence as we go along. You can always tell an insertion because it's a little bit longer, one or two letters. A deletion, it's a little bit shorter. So as you add and switch these bases around, you can actually do some damage to an organism. Okay. So again, what happens here, and I know this picture is a little difficult to understand, you're changing the sequence of amino acids. If these are amino acids here, met-lyse is the same, but look at these two. Those, the phi and the gly, turn to lu and ala. So mutations can change the amino acids in a protein, which can change the whole protein. All right, let me give you some examples of mutations here. This is a classic example. All right. Here is a normal red blood cell, and here is what's called a sickle cell or a sickle red blood cell. It's kind of shaped like a moon, a half moon. Now, only one single letter change, a single base change, changes the shape of this cell. Now, why can this be dangerous? Well, these cells, they don't flow well around the body in the bloodstream. They get kind of stuck. They all kind of clump together. They all kind of hook up together, and that can cause blood clots, and that can rob oxygen from certain organs. It can be deadly. This can be a deadly disease if it's not treated. This is a genetic disease caused by a mutation in only one letter. One letter in that DNA causes this change. Okay, so this is an example of a bad mutation. Here's another bad mutation. It's called cystic fibrosis. And what happens, as you can see here, the normal sequence of letters, they have lowercase letters in this case. All right. Doesn't matter if it's lowercase or uppercase. Doesn't matter. You can see this ATC is deleted here. This is a deletion mutation where it deletes an entire amino acid, the code for an entire amino acid. Now, you might be saying, well, what the hell? Just one amino acid. One amino acid change can change an entire protein. And what happens is that this protein in the lungs, this is a disease of the lungs, is a defective protein. And that creates unusually thick and sticky mucus in the lungs, and it blocks your airways. And oftentimes, cystic fibrosis patients have to get this mucus removed. There are other remedies as well, but it can be deadly. It can shorten your lifespan if not treated. Another bad mutation. Okay, some mutations don't cause any change in the order of amino acids in a protein. So if you switch from an A to a T or a G to a C or a T to an A, it doesn't matter. If it doesn't affect the amino acid, it's not going to affect the protein. So most mutations, I'd have to say, do not cause a problem. But other mutations can change the order of amino acids in a protein and therefore change the shape. Think about antibodies, enzymes, hormones, receptors. Those are all proteins. And if you change their shape, they don't work anymore. Okay, so mutations are bad when they change the shape of a protein. Remember that DNA codes for proteins. All right. Okay, so to kind of sum up, mutations can be harmful, genetic diseases, but they can also be beneficial. We talked about the black jaguar. We talked about the garter snake. We talked about the opposable thumb. There are plenty of examples of good mutations out there, but others have no effect on proteins. So, as you're answering the questions, the Castle Learning questions, just bear in mind that um, these mutations change the sequence of DNA. They can be good, they can be bad. There are three types: the deletion, the insertion, and the substitution. Those are the three types you need to know. And you need to know if there's a mutation, they can lead to changes in a protein because, after all, DNA does code for proteins. All right, let's wrap it up here. Again, I hope everyone's well. Please message me on Remind if you have any questions. Um, I am being very generous with the grading. So please, please, I implore you to do as much work as you possibly can. And we are, like I said, we are ending a little bit early this year with the school year. I hope to send you out a video message with some other important information, and just so you can see my beautiful face. And yeah, that's that. Okay. So good to see you guys metaphysically, and I'll talk to you soon.